Okay, so are we ready now?
No, yeah, the stream takes a good minute to warm up.
Okay.
Okay, it's quicker
Are
than
you
usual.
recording this?
Yes, yeah, there we go. Right. Good morning, everyone. Welcome to session 10 of BGA24. Today we have Ksenia from the Tree of Life Assembly team, who's going to be running through the genome assembly pipeline that we run. Take it away, Ksenia.
Okay.
Oh, need to stop sharing, don't I? Again.
Can you see my slides? So hello, everyone. My name is Ksenia, and I work in the Tree of Life Assembly team at Sanger Institute. In this session, we are going to talk about the genome assembly, quality control checks, and how assembly process is streamlined for the Tree of Life project in Sanger. Modern technologies can only produce DNA sequence of a limited size from 100 base pairs to a megabase. The whole genome has to be inferred from the millions or billions of reads. This causes a problem of genome assembly. In this session, we will focus on assemblies of the data obtained with the whole shotgun approach, as the most widespread nowadays. It is based on fragmenting the whole genome into small pieces, which are further sequenced. The individual reads coming from all the different fragments of the genome are processed together to make a full genome assembly. Before we go further, let's define some terms that will be used throughout this session. A genome is a complete set of genetic information that makes up an organism and is made up of DNA. A sequencing read represents an inferred sequence of genomic fragments. In the process of genome assembly, reads are combined together into contiguous sequences using their overlaps. Such contiguous sequences are called contigs. They can link together in an order and with gaps separating the consequential contigs. Such an ordered sequence of contigs is called a scaffold. It is possible to make an analogy between the genome assembly and solving a jigsaw puzzle, where reads are puzzle pieces and the genome assembly is a full puzzle. The jigsaw puzzle analogy can help us to reason what could help to produce a better assembly. Larger pieces or longer reads would mean longer stretches of continuous input data. As much as pieces can be frayed, reads can have errors that lead to the noise in the data. When we assemble a puzzle, multiple areas of the picture can look very similar, which makes it difficult to say for a piece which area it comes from. Same for genomic repeats. Repetitive sequences are one of the main reasons why the context separates from each other in their ends. Read coverage distribution along the genome is another factor that affects the results. This can depend on the technological biases as well as biological properties of DNA. As an example, depending on the type of the sequence, some areas of heterochromatin can be more difficult to sequence. A reference image can help when assembling the puzzle. The same way, a closely related genome can be used as a guide for assembling a newer data. Also, we can imagine two or more boxes of puzzles representing slightly or significantly different images. If we deal with both sets of pieces at the same time, it will add an extra factor of complexity, because now we would need to be able to tell for a given piece which one of the sets it came from. Same when we have to deal with more than one haplotype in the data. In cases of extreme heterozygosity, it can be hard to tell apart the haplotypes. Following the previous considerations, we can now line out multiple factors that affect assembly complexity. Error rate and read length are determined by sequencing technology. Also, depending on the genome complexity, it affects the minimum required sequencing depth. Illumina reads with error rate about 1% and relatively short read length, 150 base pairs, are rarely used now. With the latest advancements, long read technologies have become widespread. Where previously read depth 50x per haplotype was required, now 15x per haplotype can be enough. Genome size is an important factor in the assembly. It can vary a lot in different phylums. The biggest genome we have dealt with in total so far was the mistletoe that had a haploid size 94 gigabases. The organelles are part of full genome assembly. There can be difficulties in assembling them because of the repeats and heteroplasmy. This is when multiple different copies of organelles are present in the genome. For mitochondria, reads coming from the historical nuclear inserts called nonce also add noise into the data. And depending on the used sequencing approach, organelles might not always sequence well. Assemblies of big enough genomes, like many of protists, fungi, plants, and animalia species, require significant computational resources, which means computing facilities like servers and clusters are required to run an assembly. For raw assembly, you may need from 70 100 GB to 300 GB RAM for a genome and multiple CPUs. Small genomes of bacteria and viruses usually can be assembled on a modern laptop. Mhm. Sequencing costs have been constantly decreasing. The initial draft of the human genome achieved through Sanger sequencing had a cost of $3 billion and took over a decade to complete. Now, with the NGS platforms, billions of reads can be produced in a single run within a few hours or days, and a high-quality full human genome assembly will take a sum of several hundreds of dollars. PacBio, Oxford Nanopore, and Illumina are one of the most widespread sequencing technologies nowadays. Maximum throughput, read length, and accuracy may vary for different systems. There is a recent review on the slide showing that most approaches are capable of achieving more than 99 accuracy with a varied read length. By combining different types of sequencing data, it is possible to achieve better results in assembly. We will now go through some sequencing technologies. PacBio sequencing started in the late 2010s. As for now, there are two types of PacBio sequencing data: CLR, which stands for continuous long read, and HiFi, high fidelity mode, introduced in 2019. A sequencing process occurs inside small wells on a small microchip called SMRT cell, which contains millions of these tiny wells. HiFi sequencing uses fluorescent light signals to identify DNA bases, well, pretty much as well as CLR sequencing. A HiFi read is obtained from multiple rounds of CLR reading through a single DNA molecule and finding a consensus sequence from these reads. While CLR is relatively high error prone, 10%, with reads N50 is 20-35 kb long. HiFi read precision is 99.9%, with N50 read length 10 to 20 kb. The high accuracy combined with long read length in the HiFi mode makes this data very good for assembly. However, this technology is on the high end of per base cost. Oxford Nanopore is another widespread long-read technology. It offers a variety of devices and sequencing modes. All Oxford Nanopore sequencing devices use flow cells, which contain an array of tiny holes called nanopores. Each nanopore connects to a sensor chip, which measures the electric current. When the molecule passes through the nanopore, the current is disrupted to produce a characteristic squiggle. The squiggle is then decoded using base calling algorithms to determine the DNA or RNA sequence in real time. This technology is capable of producing longer reads. The ultra-long sequencing mode allows to obtain read set with N50 100 kb. Limitations of this technology come from the higher error rate. However, there are very effective approaches to correct the errors. Also, for ultra-long mode, there are high input DNA requirements. Hi-C is a proximity ligation approach. It captures the organizational structure of chromatin in 3 dimensions, where genomic sequences that are removed from each other in linear distance can be closer in the 3D space. Globally, most Hi-C links are on the same chromosome, and more are close and distant. Hi-C libraries are usually sequenced using Illumina pair-end technology. The pore-C is sequenced with Oxford nanopores. The Hi-C data can be used for haplotype phasing and in order to link together the context into scaffolds. Manual creation of an assembly is facilitated with Hi-C maps. A Hi-C map is a square matrix is on the slide on the right corner. Both axes represent the positions along sequences of the assembly. Each point on the Hi-C map represents a density of Hi-C signal between the corresponding position along the assembly. The information can be used to find and correct different types of misassemblies. Let's now have a look at the fundamentals of the assembly approach. Assembly process can include multiple steps depending on the genome properties, such as ploidy, and available types of reads. However, the first step in every assembly is context generation. There are two basic approaches to do that. Overlap layout consensus approach is where all the reads in a dataset are compared against each other in the search for overlaps of arbitrary lengths. The overlaps among reads can be regarded as edges in an assembly graph. Paths in such graph represent larger continuous genomic fragments called contexts. Because of all-against-all comparison, this approach is computationally expensive. It is implemented in multiple software such as HiFiasm and HiCanoe. The Bruin graph approach operates over k-mers, subsequences of length k. Usually k is not very big, such as 21 base pairs. It can be also 31 or 51. This approach took over popularity when the short-read Illumina data was most popular. Thanks to the fixed length of the overlaps, it is less computationally demanding. Because of the shorter sequences used for graph construction, it can result in more complex graph structures and, as a result, more fragmented contexts. In the latest assemblers, it can be further made up using the long-range information available from the long read if they are used as the input source. Among the long-read assemblers, Verco and LGA employ this approach. Now we can get back to the factors that can affect the assembly generation. Let's, as an example, take a genomic repeat shown on the picture and see how the region might look in an assembly graph. If the repeat length is bigger than the length of the read spanning the area, it will result in a structure where the unique sequences linking the repeats on both sides will form distinct graph edges. So here is a repeat, and here is the flanking sequences. So in the graph traversal, it wouldn't be possible to tell which of the incoming edge corresponds to which of the outgoing edge. Like we can go this way, or we can go this way. A long-range information, such as Hi-C or ultra-long read, could help find it out. Like in this case, we can think of these markers as a long-range information. So if we have two matching markers here, we can go this way. And if we have two matching markers here, we can go this way. So a long-range information such as Hi-C could help to find it out. And if it's not available, we will get 4 contexts, like separately. Based on experience, better assemblies come out from the combination of multiple data sources. And in this process, several assembly stages can be applied, including the final step of manual curation. And this is a diagram of the tree-of-life genome generation workflow, starting from a sample acquisition and ending with the data release. When a sample is collected and morphologically identified, the species barcode, such as COX-1 gene or ITS region, is verified in the barcode database. Then the data, along with the metadata, is passed along the system for DNA and RNA extraction. Normally, for each genome, Hi-Fi reads, Hi-C, and RNA-seq are generated. RNA-seq are used further down the pipeline for gene annotation. The produced data has to pass QC checks, such as barcode checks, contamination screening, camera analysis, and we're going to look into it in more detail in a minute. The QC data is then being assembled in different stages. First, raw contexts are generated, then they are scaffolded up using Hi-C data. Organelles assemblies are generated separately. This process is automated, but if the automatically produced assembly doesn't look good, some work might be needed to switch parameters and look into the assembly in more detail. Once the draft assembly is good enough, it goes through the decontamination process and further handed over into manual curation. After curation, genome is released in the public databases, but the work doesn't end here. The genes are annotated using RNA-seq data at this stage. And lastly, a short article describing the assembly is released. Here, I would like to show you an example of a genome node, which is a short data article that briefly describes the species of the released genome, the assembly statistics, and all the steps that the data has gone through to obtain an assembly. It is possible to cite, and whoever uses data in their research is encouraged to do that. TOL collaborates widely on many projects, and for many of them, data is generated and assembled here. Now let's have a closer look into the data quality control. We will go through the QC steps that are performed for the TOL data. The basic approach must be similar for any other whole shotgun sequencing data. When the sequencing data comes into the pipeline, it is important to confirm that the dataset indeed belongs to the right species. With this purpose, a schemed subset of the reads is mapped against BOLD database. BOLD database is an online community database keeping barcodes for millions of species. The species identification happens based on the COX-1 gene located in mitochondria. For many vertebrate and invertebrate genomes, BOLD provides precise species identification. UNITE is another online resource that matches a barcode against a curated set of resources. It can be very useful for fungal genomes. For fungi identification, it is recommended to use the ITS gene in nuclear DNA. Normally, read data for each type of sequencing goes through the same barcoding checks. However, there is a caveat. For Hi-Fi, a read can cover the whole COX-1 gene. Hi-C reads are 150 base pairs length, which means that the only fraction of COX-1 gene can be captured by one read. This may cause ambiguities that have to be investigated. Apart from the barcode checks, Hi-Fi data is evaluated by other qualities. Yield and distribution of read length characterize the genome coverage and quality of sequencing run. Hi-Fi data has to be also screened and filtered from barcodes and, depending on the protocol, from PCR adapters. K-mer plots can be used to evaluate the quality of the raw data. Here is the K-mer coverage distribution plot generated by GenomeScope. The K-mer coverage are on the horizontal axis. The vertical axis corresponds to the K-mer frequency. A GenomeScope profile for a deployed genome can look like this one on the slide. The blue area on the graph is the distribution observed in the data. The first peak corresponds to heterozygous K-mers. The second peak corresponds to homozygous K-mers. The high frequency, low coverage area indicates that what is most likely errors in the data. Small high coverage peak here is most likely coming from genomic duplications or repetitive regions. The black line shows how, it shows a predicted model, and model is being predicted using the parameters deduced from the incoming data. So in this case, it shows that the observed data match the model very well. The relative height of haploid and diploid K-mers reflects the genome heterozygosity. Lower heterozygosity levels corresponds to the decrease of the height of heterozygous peak. GenomeScope provides estimates of heterozygosity and for genome size, which can be useful for assembly quality control. Like, you can find them in the header of the picture. Here is a couple of examples for the different ploidy. It has to be noted that ploidy value is passed to the GenomeScope as input parameter. The figure on the left corresponds to an attempt to build a diploid profile from the haploid data. We know that male hymenopterous are usually haploid, while the females are diploid. When the incoming metadata lacks information about sex, we have to figure the ploidy out on the go. So in this case, the genome looks haploid. The figure on the right corresponds to the K-mer profile for a triploid beetle. So we can see here equally distant peaks of 1x, this is 2x, and 3x coverage. Another notable example is when the sample data contains cobions or contaminants. The plot on the left corresponds to a hi-fi data set of a mushroom that was contaminated by another mushroom of the same genus. And the left peak is an indication that there was an admixture. The figure on the right corresponds to a different sample of the same species as in the previous plot, but without admixture of the close species. So you can still tell that there is some cobionts in the data, but it's not as defined as on the plot on the left. Smudge plot is another method that can be helpful in data QC. Using statistics derived from k-mer coverage, smudge plot estimates genome ploidy and provides a heat map plot, helping to assess this estimate. I believe there is a smudge plot session in BGA, so I'm sure everybody is welcome to join it. For the Illumina high-C data, the common metrics such as yield and barcode checks are important. Apart from them, there are estimates on mapping rate, PCR duplication rate, ratio of reads mapping to multiple locations, and fragment separation. Very low mapping rate can be indication that sequencing experiment went wrong. There is an example of it in the image on the left for the data with a very high PCR duplication rate. Otherwise, it can be an indication of sample slot, which what could have happened here. Fragment separation is an important metric as it defines how well scaffolding can work. This estimate can be obtained by mapping high-C reads onto the subset of largest contigs in the draft assembly. In the plot on the left, the horizontal axis is the insert size of the pair end reads. The frequency of these values are on the vertical axis. The dark blue line shows the insert site density measured relatively to the mapping of the contigs. We are particularly interested here in this shape in the long range distance because we use the high-C data to join separated fragments. So ideally, this should be a curved shape. So the plot on the left is an example of a good high-C data because the shape of this area is curved. And you can tell it because, so this is an example of bad high-C data. So this is a more concave, you can tell, distribution, which tells that the long range distance is not really well present in the data. And now we can go through the steps of the tree of life assembly pipelines. The goal for each genome in TOL is to provide an assembly with a complete curated representation of one single haplotype. The sequence of alternative haplotypes are usually released as a satellite assembly. There are two approaches to generate draft assembly. In primary assembly, sequences can be more complete and contiguous comparing to the sequences of the alternative haplotypes separated in a different file. However, there is another approach, which contains and generating fully phased haplotypes in which either of assembled haplotypes is supposed to be a full representation of the physical ones. Just not like one with satellite sequences in the primary, but just all the haplotypes are fully phased and fully assembled. In order to produce such an assembly, we use the high-C data coming from the same individual, or alternatively, trio information can be used. Of course, if the genome is haploid, there is no alternative haplotypes. And for each assembly, organelles such as mitochondria and chloroplasts are identified and assembled separately. We can now have a look at the so-called pre-alt primary alternative assembly in more detail. First, hi-fi asynchrone assembler is run on the raw hi-fi reads, which produces primary and alternative contigs. It's common that there is some retained alternative haplotype in the primary assembly. So the second step is to purge the primary contigs from alternative using purge tubes. The purged primary contigs are then scaffolded up using high-C data with a scaffolder called YAS. The final scaffolds and haplotics are further passed over into decontamination manual curation. For a fully phased assembly, hi-fi is run in a high-C phasing mode. Then both haplotypes are scaffolded up. The two fully phased haplotypes can be combined for manual creation to fix misassemblies, possibly remove remaining bits of remaining haplotypes, and for chromosome annotation. Contigs or reads coming from organelles can be spotted using reference organelles assemblies. In the public databases such as NCBI, there are loads of reference assemblies for vertebrates and vertebrates, plants, and this approach is implemented in MitoHiFi software. The limitation of this approach is that clades that are less studied will have poor representation. Also, unique repetitive sequences that are not captured in the reference can complicate the procedure. And in some clades, such as fungi, the size of organelles can vary even in the same genus, which also breaks the reference-based approach. OTK implements a chimer-based approach, and it is a de novo approach, which consists in building a graph from the read data first, and then scanning it for organelles using pre-trained hidden Markov models. So now we can have a look into assembly quality checks and possible ways to fix the issues when something goes wrong. Numerical statistics can help to understand if the assembly actually worked. It is important to note the assembly size and how it agrees with the estimated genome size. And the estimated genome size can come from genome score. Another important consideration is the distribution of sizes of assembly sequences. In that respect, N50 is a particularly useful measure. So N50 equals X means that the contigs of scaffolds of length more than or equal X constitute 50% of the assembly. So you can imagine like half of the assembly and the contigs covering half of the assembly, and you find the biggest length of the contigs covering this assembly, and the minimal of them will be N50. So NG50 is analogous measure relative to the genome size. L50 is a number of sequences making N50. In case scaffolding is performed, another informative metric for a scaffolded assembly is number of gaps. It can be useful to compare statistics before and after scaffolding. And useful and flexible tool for generating these statistics is GFA stats. And here on the right, there is an example output of GFA stats on a scaffolded assembly. So here the number of contigs and scaffolds are the same, and there are no gaps, which means that scaffolding didn't actually produce any joints here. Contig N50, scaffolds N50 is the same number of contigs, scaffolds. BUSCO states for benchmarking universal single copy orthologs, and it is used to estimate assembly completeness in regard with the core genes. BUSCO provides an estimate of complete, missing, and duplicated genes. Apart from completeness, it can very well highlight issues with haplotype purging. BUSCO runs against a gene set specific to the clade. That's why it's hard to compare BUSCO scores against different gene datasets. Also, BUSCO sets for some underrepresented clades can be lowish. It doesn't necessarily say assembly is bad. We have earlier discussed chimer metrics for quality control of the raw data. Similarly, there is a way to use chimers for estimating assembly quality. The approach called Mercury was originally described in the paper in 2020, but here in production, we have been using a fast implementation called Mercury FK. The assessment is usually run against a read dataset. It's possible to use reads used to produce the assembly or some other high-quality dataset of reads. It can be useful for assembly from error-prone long reads against a dataset of high-precision short read sequencing. There are two useful quantitative metrics. One of them is chimer completeness, shows how much of the different unique chimers present in reads is also present in the assembly. So it's a ratio metric. For HiFi assemblies, we consider 98, 95, 99% completeness as a good one. If it's getting lower, it has to be investigated. For the CLR or ONT assemblies, depending on base calling quality, this ratio can be lower, like 95%. Another useful metric is an assembly QV, which helps to assess base quality. This metric can be regarded as a score and doesn't have a measuring unit. For hi-fi assemblies, we usually aim for QV60 and higher. Next, similarly to genome scope profile for reads, there is a k-mer profile for an assembly. Copy number plots show k-mer representation against the read dataset. And there are also spectra ASM plots. They can compare k-mer representation between two assembled haplotypes. Here is an example of k-mer copy number plot for a deployed assembly. Horizontal axis is k-mer multiplicity in read data, and vertical axis is k-mer count in read data. Color shows counts of the corresponding k-mers in the assembly. So the scales are pretty much the same as in genome scope. It's just the color that helps us to understand how well assembly went. Black-colored k-mers appear only in reads. Red-colored k-mers appear in assembly only once. And there are blue-colored k-mers that appear twice, and so on. And there is a legend that tells us. On this example, the figure on the left corresponds to the assembly of one haplotype. The figure on the right corresponds to the assembly of both haplotypes. For the second plot, we can see there are heterozygous k-mers that appear either in one or another haplotype. Diploid or homozygous k-mers appear in both haplotypes twice. Half of the heterozygous k-mers on the left figure lag from the assembly, and they will appear in the second haplotype. And you can tell that they will appear in the second haplotype because on the joint plot, they both are present. All deployed k-mers in the second peak are present in one assembly, in one copy. Yeah. The relative height of the black area and the red area for the heterozygous k-mers can reflect issues of the haplotype separation. Ideally, we expect high unique k-mer present in each haplotype. Here is an example of the plot for a single haplotype assembly, which shows signs of retention of the second haplotype. So this is a blue area. But also at the same time, we can see that it was a little bit over-paging because there is a black area in the homozygous, under the homozygous peak. In our pipeline, haplotype sorting is done first by hi-fiasm, and in case of pre-alt assembly, by page dupes. Here is an example of how some deployed k-mers replaced by hi-fiasm on the left are further being paged by page dupes, and this is what happens. So in this case, actually, there was, although the retained haplotype was removed, there is still a bit of over-paging there, which has to be sorted. Both hi-fiasm and page dupes operate by estimating read coverage along the context and introducing haploid deployed area thresholds based on haploid and deployed peaks. Figure on the left illustrates correct coverage thresholds for hi-fiasm. It's a known issue that for highly heterozygous genomes, hi-fiasm may misidentify heterozygous peaks for the homozygous, and it can be fixed by setting the manual cutoffs. Like this is actually what happened here. The heterozygous peak was taken for homozygous, and this is actually the homozygous peak, but it was not noticed by hi-fiasm. So there are some other considerations to use when assessing haplotype separation. In some situations, without running assembly, it's hard to tell the genome ploidy. A single peak with noticeable overpaging is an indication of the haploid assembly. Equally, a genome can be polyploid, which can cause confusion for the tools not designed to handle multiple ploidy. In case page dupes were run, it's worth checking what sequences were paged and for what reason. And for that, you can have a look at the output files. Some very repetitive genomes will have a small third peak with multiple k-mers, which is just an indication of its repetitiveness and doesn't have to be paged. If haplotypes don't separate very well computationally, it can be possible to create a joint Hi-C map and try to separate context based on the Hi-C signal. We have briefly mentioned HiC-map in respect to the data QC. HiC maps allow to inspect the density of HiC signal among different parts of the assembly. It can be a very resourceful tool for assembly QC and further manual curation. Here is an example of a nice HiC map. High saturation and uniformity of one of the signal along the sequences in combination with a very low off-diagonal signal is an almost ideal example of clean assembly. On the HiC map on the slide, there is an off-diagonal signal between two sequences here. It allows to tell that these two sequences belong together and can be manually joined. And there is a special software to deal with it called pretext view. Let's have a look at some quality metrics for a beetle genome. A slight retention of diploid k-mers is visible here on k-mer plot, which is also reflected in an elevated Busco duplication rate. However, completeness is very nice, like 99.4%, which is good. A HiC map for this assembly contains a fair amount of small shrapnel sequences, which actually can be indicative of multiple things. It could be retained haplotype or signal duplications or unplaced repeats or contaminated sequence. It's possible to tell each type of the event by examining distribution of HiC signal on the HiC map and the level of Hi-Fi read coverage among the sequences. In this case, most of the shrapnel represents a retained second haplotype, and you can tell it by having a closer look at the signal between shrapnel sequences and the main chromosomes, like I just here on the right, I just zoomed in one of them. So this diagonal sequence example suggests that there is a high affinity, which means a high similarity between the shrapnel bit and the main scaffold. Like that bit is really highly saturated. For some genomes, such as marine or fungal, it is quite typical that they come along with cobionts. Here is an example of lichen Cladonia navegica. As we know, lichens are symbiotic organisms consisting of fungal and algae components. And being sequenced and assembled all together, the genomes can be hard to separate using k-mer clustering and reference-based mapping approaches, although they are still very good approaches that we do use. But in this case, it was very helpful that the genome separated very well by HiC signal distribution. The highest coverage sequences in the left corner belong to the fungus. And here is a component with slightly less HiC coverage is one algae component, and this was the other. One of the convenient ways to inspect cobionts in an assembly is by using a blob toolkit software. There are plots for two assemblies on the slides. The left slide corresponds to the assembly of the lichen we have just talked about. And the horizontal axis is GC content, and the vertical axis is the Hi-Fi read coverage. The blobs are assembly sequences scaled by the relative lengths. Each is colored according to the phylum having highest Busco score. And here the highest coverage has the fungal genome. And yeah, you can tell that, by the way, by how the other compounds are grouped, that it can be a bit tricky to separate them. The plot on the right is also for a fungi called Scurfy fiber cap. And you can tell by the wide range of GC content distribution that the data set contains quite a big range of compounds. We have discussed the nuclear assembly and can now have a look at the specifics of organelles assembly. Once mitoHiFi or OTK generates a candidate organelle quantic, it has to be looked into. Assembly size must be close to the reference organelles, or at least typical to the clade. It is important to check that the organelle known to be circular is circular. It is also helpful to run through a quick gene annotation and compare the gene content to the gene content, the reference organelle genome. For mitochondria, normally there are 37 protein coding and tRNA genes. They shouldn't contain frame shifts. It can also be possible to assess the organelle assembly by mapping Hi-Fi reads to the sequence and visually assessing the mapping consistency. Like here on the slide, there is an example of a very nice read mapping for fungal mitochondria on the right, so this is fine to go. This one on the left has very uneven coverage, even with gaps. So this definitely has to be investigated. Yeah, so here I wanted to show you an example of QC checks. For every genome in the tree of life, there is a dedicated page called TalkQC that contains the discussed data and assembly quality checks. Yeah, so this is the name of the species, which one of the Hymenopterus species. And there is some general like metadata which can be pulled from public databases. Here we've got like a range of available. It can be multiple specimens or just any samples that you have at hand. So these are all come from one individual, but from different organism parts. And importance, this one is female, which in the case of Hymenoptera would suggest that it's going to be deployed. So this is PacBio and IC data generated for this data set. Both come from the same individual, and given its haploid, it means that we'll be able to generate both primary alt and phased assemblies. But just looking at these stats, which were all gotten from the GenomeScope run, there is a haploid run. We shouldn't use IC stats for estimation. We use PacBio for estimation, like 190 megabases length and heterozygosity relatively high, 0.88. Yeah, so this is a GenomeScope profile, which is well, you can tell that it fits quite well to the model and is definitely deployed. Not that heterozygous actually, well, because you can tell it because homozygous peak is higher than heterozygous. But yeah, this is quite good to go. So this is the high line plot that I previously told you about, and high C data is quite good. So you can tell that the long range information was captured quite well here. So hopefully it will work for scaffolding. And here, like as part of QC, what is being done that the 30 longest contigs are being separated from assembly, and without scaffolding them, the high C map is created, like high C data is mapped on the contigs. And you can tell the point of this is just to see how well it maps and visually represent it. So in this case, mapping is quite good. Here was a bit of an issue because there is another like closer look in the PacBio run data. So it was run in ultra long sequencing mode, and you can tell here there is an issue because like it's red and it indicates that the barcode check mapped the reads to a wrong species, and it can sometimes happen because it just a scheme data set of reads, and it just mapped on the pulled bold database, which is not necessarily the full cover of the bold database. So in order to investigate this, we would need to run the mitochondrial assembly, like do it all and annotate the COX-1 gene, and then put the COX-1 gene into bold or to blast it and to check if it blasts to the right species. And in this case, that was all done and it blasted to the right species. So it was good to go. So this is the result of Illumina data run. So this is yield. There's also the yield for Wi-Fi data previously. And again, so barcode orange, which means that the high C data didn't really map anywhere for barcode checks. And this is exactly the issue that I previously told you about, that because Illumina reads are a bit short, it can be tricky to capture the whole COX gene. So, but in this case, like we know that Hi-Fi and Hi-C now come from the same individual, and Hi-C maps to the Hi-Fi assembly very well. So we shouldn't really be concerned about this barcoding check. So here we run a few K-MER approaches to roughly scan for cobariants, and just actually it tells us like roughly which like it just takes some context and tells us what species or what genuses they represent. So these are all Hymenoptera, that should be right. And finally, we come down to the assembly stage. So this assembly section, because we run FBOST phased and primary alt assembly, each line in this table corresponds to one of the stages of both assemblies. Like if we focus here on pre-alt, so this was run, this is the stats for the primary high-chiasm assembly. This was after it was purged, and this is after it was scaffolded. So you can tell that Busco score is good right away and without much duplication, but it was further removed after purging. And so about scaffolding, you can tell. So this is the number of contigs. This is the number of scaffolds, and scaffold on 50 versus contig on 50. And you can tell that, yeah, okay, scaffolding really worked because, well, we see that some contigs were joined and 50 increased. Yeah. Oh, yeah. While the total length like remains the same. Okay, and so this organelle section, this is a case where we would probably have to look into, because we know that Hymenopteras, because they are very, their mitochondria are very repetitive. There can be difficulties with comparing them against the assemblies of organelles from Hymenopteras present in the database. And because of that, the mitoHiFi, which is reference-based approach, doesn't always work. And in many cases, we have to run the OTK approach, which is just not listed here. So yeah, this will have to be looked into. Okay. So this is all for the quality checks. And now let's have a look at the Sanger Tree of Life assembly pipeline. The current production pipeline is implemented as a set of in-house workflows. We have been working out on the next low version of the pipeline implemented with NF Core workflow template. Eventually, we plan to switch to it in the production. The code is hosted on GitHub and free to use for the community. The current version of the pipeline implements all the basic assembly steps, starting from raw assembly, running page tubes, scaffolding, and identifying organelles. Optionally, polishing of the assembly with a 10x data can be used. The input dataset for the pipeline is described in the YAML file, which is shown on the slide. And you can refer to the pipeline docs on the GitHub page or have a look at the example files to figure out the exact format. We can go through it a little later. The file must contain paths to the all input data and some meta information, such as Busco library for QC checks and mitochondrial code for screening organelles. Yeah, so we can now have a look at the GitHub page and quickly go through the pipeline structure. Oh, sorry. The main workflow is defined in the genome assembly NF workflow file. Yeah, so if you, what it does, it imports the workflow files, which are located in a separate folder. So these are all different sub-workflows, and they are all here if you want to have a look. If you want to try and run this pipeline, you can refer to the documentation. So for that, you can go to the docs and usage MD file, just scroll down a little, and there is a way to try and run it in a test mode. Yeah, so in a test mode, you just can put assets test YAML, and for Sanger, so this will work for Sanger, but it might not work for this configuration. This profile might not work for other institutions, so feel free just to drop it. The example data sets can be checked in the assets folder. So here you have to look for the YAML files starting this test. So what you have to provide in case you want to run polishing, but you don't have to do it. So it's an optional part. So you have to provide Illuminator, but the main input is PacBio reads. In this case, they are all hosted as on a bucket, and HiC reads. But well, it's of course fine to have them just located on your file system. So it's a HiC motif which can be passed into YAS at the scaffolding stage. So HiC aligner, so you can switch between WM mem, which is default, or there could be minimap 2, which is implemented for big genomes. Also, you have to provide, if you want to run Busco, you have to provide lineage. And for mitochondria, you also have to do give some information, because we run both mitochondria and OTK. So this is the minimum length to query for the reference for mitochondria, and the species that we will look for as the closest reference, which is itself probably. So this is mitochondrial code, which is 5. Like for this data set, you can look it up for your species on NCBI. And for OTK, we also have to provide some HMM profiles. So feel free to run it. And also, I forgot another thing, that if you want to modify some parameters for software, like run FastK with different settings, or perhaps you want to run HiFi with different settings, you can, like for now, you can go into modules config, which is a standard location for this next level workflows, and to pass it like it's done here, like auths, and it will be picked up by the pipeline. So before release 1.0, which will be production release, we will still want to implement some features that are available in the current production pipeline. There will be different entry points, like you want to be able to run organelles separately, maybe scaffolding or HiC mapping also be possible to run separately just by configuring the pipeline. And apart from that, the pipeline is ready to use, and everyone is very welcome to give it a try. And this is all that I have for these questions, and I'm happy to take questions if there are any.
We have one in the Discord at the minute.
Okay.
From Capture. Hi, Ksenia. I just looked at the TOLQC page for another species where OTK was used for organelle assembly. Strangely, there are reference sequences given for the assembly, even though OTK should work de novo. So what was the given reference used for?
Well, I believe it was, so it's probably because we run OTK, but also OTK provides just mitochondrial, it provides a config, but it doesn't annotate it. So we usually run mitochondria on top of OTK. So when OTK already produces results, we want to QC them, like QC this mitochondria, like to find how many genes there are, if there are frame shifts in these genes, and that's why mitochondria is run over it. And just for that, the reference assembly is queried. So it's actually, it doesn't have to do with OTK, it's more with QC check of OTK run. Okay.
Cool, thank you. And there's one in the chat now. How did you make the HiC fragment separation plots? Was it a particular tool used?
Well, yeah, so it's actually an in-house tool. I'm not sure if actually it's available outside. But outside of Sanger. But it's just an in-house highline thing. Actually, I could try and have a look if it's. Yeah. Okay. Yeah, it should be available for public. So to be honest, it's a bit tricky to run. So it might be just easier to implement something of your own. But if you want to do it, I can put this web link into into the chat. Okay. Yeah, feel free to use it, but be prepared that it's a bit can be a bit tricky to run. Yeah.
Any more questions?
Okay, seems all good.
Yeah, deafening silence.
Right, shall we wrap up then?
Yeah. Yeah, so if there's no more questions, thank you everyone for attending. Thank you, Ksenia, for giving it your time to give this session.
Yeah,
And
thanks everyone for coming. Hope
and
it was useful. Okay, thank you all. Thank you, Damon, for
cool.
your effort in organizing it.
Thank you. All right, I'll see everyone in the next session then. Goodbye.
See you. Bye.